many. There are numbers here 4, 5, 7,
Yeah,
and I don't know if there is a
I'm a
gradation.
senior
that is
scientist.
the grade, the grade.
Okay, okay, that is
I'm a
good.
senior scientist, period.
Okay.
Yeah, now you can go.
Okay, sir.
Yeah.
Welcome to all the viewers here. Welcome to all the postgraduates and all the pediatricians on this World TB Day. Today, we know on March 24th, we are celebrating the World TB Day. Echo ಆಗ್ತಿದೆಯಾ sir?
ಇಲ್ಲ ಇಲ್ಲ ಈಗ ಆಗ್ತಿಲ್ಲ.
ಆಗ್ತಾ ಇದೆ.
Sure. Huh?
Yeah.
I could hear.
No echoing.
Okay, okay. So I welcome you all on behalf of this program is done by IAP ID chapter. Myself, I'm Dr. Naglata, Secretary of ID chapter, and Dr. Ravi Shankar sir is here, who is the President of IAP ID Karnataka chapter. We both welcome you to this platform for this wonderful webinar on this World TB Day. Program is being held by the support of our central IAP. I thank Dr. Piyush sir and Dr. Basavaraj sir for their constant support for all our endeavors. And then we have at the central, we have Dr. Yagnesh and Dr. Kaya madam. And sir, there is echo, sir.
You put one in the silent mode.
I have logged out. There is only one
Mute
device.
everybody else.
Echo ಆಗ್ತಿದ್ಯಾ?
Sir, there is an echo.
ಇಲ್ಲಪ್ಪ ಏನು ತೊಂದರೆ ಇಲ್ಲ ನೋಡಿ.
ಎಲ್ಲರೂ
ಮಾತಾಡಿ
mute ಮಾಡ್ತೀರಾ sir?
ಮ್ಯೂಟ್ ಮಾಡ್ತೀನಿ ಎಲ್ರು. I mute everybody. Yeah, now you can unmute yourself and can speak. Yeah. Unmute yourself.
Okay. This program is done by the support of Karnataka chapter also. So I welcome you all on all our behalf. Today we have a wonderful session on this World TB Day. We all know that March 24th we do commemorate. No sir, there is an echo definitely.
No, somebody has put the two things on, maybe that is the reason. But otherwise there is no echo at all. I mean, there is no noise, something is not there. ಇಲ್ಲಪ್ಪ ನೀವು ಮಾತಾಡಿ ನೋಡೋಣ. ನಂದು ಇಕೋ ಆಗ್ತಿದ್ಯಾ?
ಇಲ್ಲ ಸರ್.
ಮತ್ತೆ ಅಲ್ಲಿ ನಿಮ್ದೇ ಏನೋ ಹಾಗಾದ್ರೆ ಅಲ್ಲೇನೋ ಆಗ್ತಾ ಇದೆ.
Okay. Okay, sir. No, this day is celebrated to. This is day celebrated to create awareness among the public for about this devastating health. Also commemorates the date when in 1982 when Robert Koch announced that he had discovered the bacterium that causes tuberculosis. So we have a very eminent speaker here among us. We have always had pediatricians talk, but we have somebody who has done an original research, and we are very proud that sir has paid time to be with us and to tell us about the evolution of drug resistance in tuberculosis, which has become a menace. So I welcome Dr. Vinay Kumar Nandikoti sir. Sir is the senior staff scientist. Sir has a CV running around 7 pages. Excuse me, sir, if I miss out something in those things. Sir has been, sir's education, sir has been awarded MSc by Indian Institute of Technology in Mumbai. And sir has had from PhD from Indian Institute of Science in Bangalore. Sir has held various positions and employment, starting from as a research associate in Texas and University in USA, to this day where sir is working as a staff scientist in National Institute of Immunology, New India, in New Delhi in India. Sir has so many awards and so many academic citations and publications, innumerable publications. So the list of publication goes on. total publication 67, peer-reviewed publication around 65, sir has chaptered around 2 book chapters, review 2, and the total number of citations is approximately 2000. We are indeed very proud that you are here to enlighten us about the TB drug resistance. So we welcome you wholeheartedly from IPID chapter, and I hand over the mic to, I request Ravi Shankar sir to speak 2 words before sir begins his speech.
Yeah, thank you, Nagalata. About a few years back, I visited his lab in New Delhi. It's a central sponsored organization, and just I visited there. Somebody is, yeah, now I'm getting, yeah. And it was a fantastic lab where the research on tubercle bacilli was going on. And some of the students working under him to take the PhD, and he showed the lab everything such a fantastic way. I'm really proud of this man and proud of the institution which is doing so many things in tubercle bacilli. As a practitioners, medical practitioners probably will not be knowing about these things. What exactly the basic scientists are doing to us to know about the not only tubercle bacilli, any other microbiology system. In fact, his wife is working on leishmaniasis. So many things are coming up. We will not be knowing. They are the background. They are giving us the light, and we are following that light and treating the patient. Something like we are the pujas. The Vedas and everything is there from them. They tell us we are following these mantras and following it. It is something like that only. So fantastic way they are doing it. Really, I'm honored today because they accepted my invitation. When I suddenly remembered March 24th will be a World TB Day, then I was thinking what to do. Then I suddenly remembered Vinay. So I thought this is the best time to call him and honor him on this day. The honor the scientists in his in him, and in way we are honoring ourselves because they are the one they are giving about the light to us. So I welcome Vinay, who has done a wonderful job both in USA and in India, and to go ahead with his talk.
Thank you, Ravi. That is extremely generous introduction. So thank you for this invitation and an opportunity to share our research with people who are on the front line, essentially. You people actually deal with the patients, and you actually are the ones who are treating them. Right? There is an echo. I think everybody else needs to mute. Can everybody else mute it?
I think everybody is muted actually.
Including you.
No, what I think is somebody has opened 2 things. That there is an echoing is occurring.
I understand. If everybody else mutes it, it won't be a problem.
If I'm not wrong, I can see everybody is muted here. Let me see once again. Let me do once again then. Let me do once again.
I'll unmute myself and we'll start, including you. We'll
I will. Just 1 minute. Yeah. Yeah. Now you can unmute yourself, Vinay.
Okay. There is an echo,
Okay.
but hopefully it will be better.
Yeah, yeah.
So, since it is a World TB Day, I want to start with actually showing you the news piece where Robert Koch's discovery has been announced to the world. You can see that. That is Robert Koch and his discovery of tuberculosis bacilli. And you see the date on it, March 24, 1982. So it's an honor and a privilege to deliver any talk on tuberculosis, and it is important to actually remember him. He's the one who's actually started it all. So my talk is on evolution of drug resistance. I'm going to give a research perspective, not exactly, I will start with the basics. I'll try to explain the fundamental concepts and eventually talk about some of our most recent research that is happening in this area. So the work, before I start, I don't do the work on bench. My students do. So this work is done by this girl, Sabanas, who's been with me for the last 6 years. And she's now almost at the end of her PhD, and she will move on for some place abroad for postdoc. And this is done in collaboration with my PhD mentor, Umesh Varshney, with whom I have done PhD in IISc Bangalore. And there's a guy, a good friend of mine, Dheeraj Kumar at ICJB, and his people. Yogendra Singh, a long-standing collaborator from Delhi University. And these are my students, Satyanarayana Nagarajan, Divya Arora are my students. Obviously, nothing can be done without funding from various sources. The funding for this comes from NIA New Delhi and from DBT New Delhi. Mycobacterium tuberculosis is a pathogen that grows extremely slowly when in vitro, in the sense that if you want to culture this bacteria, you see, I'll tell you comparison's sake, microbiology is quite an easy thing to do. If you take a E. coli and put it in the culture, in about next 12 hours, you will have a fully grown, heavily grown E. coli culture. You do the same thing with Mycobacterium tuberculosis, it takes 21 days to grow. Not 1 or 2, 21 days to grow. And moreover, Mycobacterium tuberculosis cannot be in the laboratories. We cannot work inside a laboratory. We have to work in what you call as a negative pressure BSL-3 facility. See, it's one thing when you are in a hospital, you are exposed to this pathogen because of the patients. That is understandable because there is no option there. But when you want to work on the pathogen, it is categorized as a BSL-3 pathogen, and one has to work in a BSL-3 facility because working is not exactly what you call as a job in the job description, in the sense that you have to protect the people who work on this. So Mycobacterium tuberculosis is an ancient pathogen. You go back in mummies, you'll find them. And you'll find them, and it's like it claims almost 1.5 million lives per year. Right? And emergence of multi-drug resistance and extensively drug-resistant TB poses an immense threat to control the disease. Right? India has the reason why it is an important organism for anybody in India to work is India does have the highest burden of TB in the world, with an estimated 2 million cases annually, accounting for approximately 1/5 of the global incidence. So what I would like to state is that as I already said, that if you want to work with Mycobacterium tuberculosis, you have to work in a specialized facility, wearing all the suits, which is now, I think people kind of got used to it because of COVID. And it used to be the biggest killer before COVID came along. And I'm sure once the COVID goes away, tuberculosis is still not going to go away. So the tuberculosis is transmitted from an infected individual to a healthy individual through aerosols. So masks will protect us even from tuberculosis in addition to COVID. So when you actually are traveling in a public transport, somebody sneezes, and assuming they have infected with TB, they are going to give out aerosolic droplets containing this bacteria, which are taken up by us. So now once it gets taken up, the first thing that happens is your immune system comes into play. There is something called innate immune response, where macrophages are the primary cells that are involved in innate immune response. They take up the bacteria, and the bacteria it has the ability to survive in this innate immune cells, though they are supposed to kill them and present them to T cells and other cells. They survive in the phagosomes, but anyway, immune reaction goes on, and what happens is the infected macrophages where the bacteria is replicating is immediately surrounded by multiple other immune cells, including neutrophils, lymphocytes, and all these things. So it's kind of a walling off of the bacteria.
Sir, we cannot see your slides, sir. I think we just lost.
Your slide went up. One second, you start sharing it. Now, what happened? Probably you clicked on this one, so it may be in this side. So you have to click on the PPT, then it will come there to the main menu. Correct. Yeah.
Okay. Yeah. Can you see?
Yeah, now we can see.
So once it is inside, when the infected cells are then walled off with other immune cells, forming a classical structure, what all of you would know as granuloma. So this granuloma is what we call as a ceasefire situation, where the bacteria is surviving but not thriving. Your host is contained the bacteria but not out of threat. Such a granuloma has 3 different fates that can happen with them. One is that eradication of bacteria altogether. Your immune system is strong, you eradicated it. That's such a granuloma is called calcified granuloma. And in the other one is an active disease where bacteria won the battle and it starts spreading or breaking the granuloma and comes out of it, infects the neighborhood lung tissues, and eventually spreads to other people through aerosolic infection. The third one is what happens in more frequently in 90% of the cases. What happens is latent tuberculosis infection, where granuloma is there, tuberculosis is alive but not replicating, and this thing stays dormant for ages, forever actually. When the situation becomes better, for example, one gets an HIV infection, or suppose one ends up getting, you start taking anti-inflammatory medicines because of rheumatoid arthritis or something else, inflammatory bowel disease, any of these things, you need to take anti-inflammatory drugs. When you take that, your immune response, the inflammatory immune response goes down. Under those circumstances, latent TB can get reactivated. So if it gets reactivated, you end up getting active TB disease. So these are some of the problems associated with TB. So what do we do when we go to the doctor with all the symptoms, which we come to you with all the symptoms like, I have no appetite, fever that doesn't go away, weight loss, etc., etc. Then based on that and based on the few detection techniques such as using the PCR, one figures out that it is TB. Once it is considered TB, they are given anti-TB drugs where a combination of 4 drugs are given initially, eventually with combination of 2 drugs. So there are 4 drugs that are considered first-line anti-TB drugs: Isoniazid, Rifampsin, pyrazinamide, ethambutol. So these are the drugs that are used, and this therapy goes on for approximately 6 months. The problem with this kind of an therapy is that if patient is compliant and takes regularly these medicines, they will be fine in about 6 months. But patient compliance is always an issue, which all of you know. Sometimes when they do not comply with the whole course, they end up developing resistance. So when they develop resistance, one has to give second-line anti-TB drugs for this, which are fluoroquinolones, injectable drugs. They are more toxic. And sometimes if you end up getting an XD or a drug-resistant tuberculosis, the treatments are much longer, can go up to 18 months, right? And till most of these drugs have been identified way back in 1970, and only 2 drugs have been added in the recent past to that list. They are bedaquiline and delaminid. So other than this, all the drugs that are currently given for tuberculosis go all the way back to 70s and 80s. So what is a drug resistance? See, what is a sensitive tuberculosis? Any tuberculosis that can be eliminated or not, that is not resistant to any of the antibiotics, is a drug sensitive TB. If it is resistant to one antibiotic, it is considered drug resistant, right? If it is resistant to both the antibiotics, it is considered, that is first line antibiotics, rifampicin and isoniazid, it is considered multi-drug resistant tuberculosis. In addition to that, if they are resistant to one of the second line drugs, then it is considered extremely drug resistant tuberculosis. And under those circumstances, only treatment left is bedaquiline and delaminide, right? So this is how the drug resistant tuberculosis is currently being treated. So here are some other statistics. If you have a drug susceptible TB, the chances of getting cured is approximately, successfully treating is approximately 83%. As it becomes multi-drug resistant tuberculosis, the treatment success goes down to 54. And if it is extensively drug resistant, XDR TB, you can see it's approximately 30%. So the MDR TB cases are quite a lot, 0.3 million cases per India. India, with highest burden of MDR TB, globally contributes to approximately 27% of MDR. That is what comes from India alone in 2019. This was. So obviously these numbers and the TB emphasis has gone down in the past 1 year because of our global epidemic caused by the COVID, SARS-CoV-2. But besides that, so this threat doesn't go away, and we have to continuously work. So now you have antibiotic resistance. How does one get antibiotic resistance? Antibiotics, basically what they do is they enter your cell, followed by enter the bacterial cell. Once it enters the bacterial cell, they target various processes. Some of them, for example, isoniazid targets cell wall synthesis, and rifampsin targets transcription of DNA. And similarly, ciprofloxacin, second line one, targets the DNA guidase, which is involved in replication and transcription of DNA. So, and then there are others like ethanbutyl targets the cell wall enzymes, and pyrazinamide targets one of the other things. So basically, there are multiple targets for each. There are different targets for each antibiotic. But then, how do you develop resistance? You develop resistance if you have changes in one of these things. You either modify the target, that is, whatever they are targeting, you modify that, or you change the metabolism of the drug, or you make sure that your cell does not retain, the bacterial cell does not retain enough antibiotics by either pumping it out or not allowing it to come in. In both cases, you will end up developing drug resistance. I will tell you about some of the mechanisms that are used, or some of the mechanisms that are known and are responsible for drug resistance. For example, isoniazid is converted from a prodrug to a active drug, which is, you can very clearly see, this is the active drug, by an enzyme called CATG. This active drug then acts on another enzyme called INHA, which is involved in the cell wall synthesis, and this forms a adduct with NAD and making this isonetyl NADH, and that inhibits the enzyme. And this inhibits the cell wall synthesis, eventually killing the pathogen. However, if there is a mutation either in CATG or in INHA, both of these things result in CATG mutation results in incomplete processing of the isoniazid, and a mutation in INH makes it resistant to this particular antibiotic. In both cases, you end up developing resistance. So, in fact, it is one of the easiest resistances that is acquired with isoniazid resistance. The other one is rifampsin. Rifampsin works on RNA polymerase, right? If you modify the RNA polymerase, it is essentially works on Rho. If you modify the RNA polymerase by mutating in some of these amino acids which are targeted by the antibiotic, it actually results in resistance because the antibiotic cannot anymore act on its target. So you can also talk about another one is called ofloxacin, which is works on gyrase. Here, the modification of the drug target, if you modify the drug target, it results in this antibiotic does not work anymore on the drug target, that is gyrase here, and essentially the inhibition is not going to happen anymore. So in addition to these things, you can also have other things such as the drug in itself is degraded by something else, which is like, for example, aminoglycosides are degraded by aminoglycoside phosphotransferases. So the intracellular concentration of the drug goes down because bacteria has somehow found a way to degrade the drug and thereby becoming resistant to the pathogen. And there are two other pathways. This one is that you have drug outside and the drug needs to be taken into the cell. If you avoid doing that, then also, that is, if you pumping into the cell goes down, that also decreases the concentration of the thing. Alternatively, whatever drug is there inside the cell, you actively pump it out through an efflux pump. In both cases, the drug intracellular concentration of the drug goes down. So these are transcriptional upregulation of some of these genes. Ultimately, all these 4 or 5 mechanisms are heavily used by the bacteria to develop drug resistance. So we were interested in a scientific question, what are the other mechanisms? And we are not the only ones. There are multiple groups across the world which are working on what are the other mechanisms that are responsible for the evolution of drug resistance, because all the existing mechanisms cannot explain approximately 20 to 30% cases where none of the known mutations in the genome occur. So there are some unknown mechanisms, and so this is an ongoing research. So how do you actually identify novel mechanisms that are responsible for drug resistance? So now you would know that we have reached an era where sequencing a genome is not very expensive. For example, I'll tell you as an example. Nowadays, people talk about personalized medicine, or you want to identify rare diseases, because majority of the rare diseases that are present in the humans are due to genetic changes that happen to them, that happen to their somewhere in their genes. So one of the ways which majority of these people are driving towards is genome sequencing. So you can sequence a genome and identify what are the mutations that are present in your genome, and then you can figure out if you have any rare diseases. Now, on the other hand, you can use the same technology for bacteria. What you can do is you can take bunch of strains which are sensitive, bunch of strains which are resistant coming from the patients, and sequence them, sequence their genome. So this is called whole genome sequencing. To give you guys an kind of a glimpse, let me tell you that if I want to sequence Mycobacterium tuberculosis, which has approximately 4 into 10 to the power of 6 bases in its genome, it costs me approximately 10,000 rupees. That's that. It does not cost more than that anymore to sequence whole bacterial genome. So essentially, people have been extensively using this as a technique. You can see that there are any strain. So anytime you isolate a strain from a patient, then you can kind of categorize it into either a drug susceptible strain, multi-drug resistant strain, or extensively drug resistant strain based on their antibiotic resistance profiles. So once this is done, basically if you sequence, then you can say, okay, I have sequenced so many drug susceptible strains, I've sequenced so many multi-drug resistant strains, and so many extensively drug resistant strains. And then what you do is you compare the sequences of this, what you got, genome sequence of drug susceptible strains with that of MDRs and XDRs, and look for changes that are there, are the mutations that are present in multi-drug resistant strain but are not present in the susceptible strain. This whole thing is called GWAS analysis, genome-wide analysis for identifying novel traits that are responsible for drug resistance. So these 2,773 clinical strain sequences that we have taken is from various parts of the world, which are all colored in red. All these sequences, we have not done anything. We simply mined the sequence that was already available in databases. So after doing all these things, we found approximately 80 genes that are 80 mutations in different genes that are only present in multiple drug resistant strains but not found in drug sensitive strains. This is called genome wide association analysis, or GWAS. So out of these 80 mutations, some of them are already known to cause drug resistance, which essentially makes us believe that our analysis is going in the right direction. But our interest is not to worry about the known mutations. We were looking for something that is novel. One thing that hit us is that among these things, we found genes that are involved in DNA repair genes, the DNA repair mechanisms. So what is DNA repair? So I'll come to the hypothesis. What is DNA repair? So for example, you go into the sun, right? Your skin, it gets the UV light. UV light results in thymine residues in your DNA to become dimers. This is called thymidine dimers. These thymidine dimers then have to be repaired, otherwise it results in a mutation in your genome. So DNA repair is an active, ongoing process. In addition to that, there is oxidative stress, there is nitrosative stress, there are lots of other stresses that you are exposed to constantly due to the food we intake or anything. Same way, it is also our defense mechanism. Means that when a bacteria enters your body, and to destroy the bacteria, our cells, macrophages, actually produce oxidative and nitrosative radicals. These oxidative and nitrosative radicals then act on the bacterial genome. So this is a way to kill the bacteria. But obviously, bacteria is beautiful, it also has to have mechanisms. So the DNA repair mechanisms that are utilized by pathogens and by us to make sure that our genetic material stays intact, right? So these mutations are corrected over a
Hello.
period of time. So there are enzymes which are responsible for taking out the damaged base, and then there are enzymes that will come and repair this mutated or the wherever you have the base has been taken out, rest of them have to be repaired. So we hypothesize that absence of DNA repair genes or mutations in the DNA repair genes may be helping in evolution of drug resistance. Why? Because our hypothesis is the following. Normally cells are regular, and what happens is when you subject them to antibiotic treatment, slowly and steadily, naturally, there is an evolution process, and some of them become resistant to the antibiotic that is given. And that is eventually resulting in propagation of that resistant strain because you continue to give antibiotic to these patients. So eventually they evolve into drug resistant strains because rest of them get killed. So what we were proposing is that sometimes when you have mutations in DNA repair genes, maybe they're helping in accumulation of mutations faster in this population, right? And that accumulation of the mutations or faster accumulation of the mutations would give them an accelerated advantage or evolution due to higher mutation rates, and eventually the evolution of drug resistance becomes faster. This hypothesis has a little bit of basis. So Beijing strain, which is known to be multiple drug resistant strain or evolves multiple drug resistant patterns very fast, actually has mutations in DNA repair genes. So to test this hypothesis, we basically went about doing, now we followed some of the things that we have identified using genome-wide association analysis on base excision repair pathway and nucleotide excision repair pathway. So these are the essential repair pathways. Do not worry too much about it. They're simply telling you that your DNA gets damaged and there are ways to repair it, and you don't repair it, you're going to suffer because you're carrying the mutations in your genome, right? So, but point is, we basically identified two different enzymes that are involved in BER and NER in our drug resistant strains. So what we did is we made a Mycobacterium tuberculosis mutant or smegmatis mutant in this, and we basically did an experiment where with the wild type and with the mutant of this particular repair thing. And then what we did is, you can see very clearly that the mutant accumulates second line. Can you see my pointer?
Yeah.
Hello?
Yes, sir.
Yeah.
Yes, sir.
Yeah.
So you can clearly see that the mutant accumulates more mutations in its DNA. But when we put back this gene, it comes back down. But what is interesting is whatever mutation that we have identified by GWAS analysis actually does not rescue this. So whatever mutation that has been accumulated in these drug-resistant strains may be contributing to higher levels of mutations in their genome, and some of them may be beneficial and are selected, is our hypothesis. This was tested for base excision repair enzymes as well as nucleotide excision repair enzymes, essentially two different pathways that are involved in DNA repair. So then, to test this in a very more significant way, from my PhD times, I've been working on something called uracil base excision repair pathway. Okay, let me tell you what happens. Our DNA is made of 4 bases: A, G, T, and C. Right? The cytosine and uracil and our RNA is made of A, G, U, and C. U is instead of tyrosine. The threonine, sorry, not threonine. It thymidine. So what happens is there is something called deamination of cytosine, which happens because of nitrite stress, H2O2. Here is the stress. And when it happens, the deamination results in cytosine residues in the DNA getting converted to uracil. So if you go back and look into the base pairing that happens in DNA, A residues base pair with T residues, G residues base pair with C residues. Now something gets mutated from C to a U will then base pair with A instead of base pairing with G. So you will end up accumulating mutations in your in the genome. So this is now we are talking about bacterial mutations. So there are two enzymes that are involved in repairing such a mutation that is identified. They are called uracil DNA glycosylase and UDGB. Both of them do the same job. So what they do is they make sure that this uracil that is present in the DNA is removed and that particular strand is then repaired eventually to make sure there is no problem with the DNA of that bacteria, right? So what we have done is that this is a Mycobacterium tuberculosis genome. You can see that it has around 4.2 megabases. That is 4.2 into 10 to the power of 6 bases present in its genome. You can see this is the whole genome sequence. So what we have done is we mutated both the uracil DNA glycosylase genes by deleting them from their native loci and replacing them with an antibiotic capsid. This is a hard procedure, but doable procedure. It really takes some level of work. Then what we did is we took a mutant strain and a wild type strain. We grew them for 15 days. And then in the presence of antibiotic or in the absence of antibiotic, we looked for the number of colonies. You can very clearly see the wild type strain where we started off with, where these mutations do not exist, does not show that much resistance. But I will only focus on this. A mutant where both the genes have been deleted shows a lot more number of colonies in the presence of antibiotic, means they have the ability to generate mutations and survive better. So this difference is approximately 100-fold, right? So you can see that clearly, not 100-fold, around 70-fold. See, when you take this particular strain, focus on this, double knockout, RV is the wild type strain. It has a certain mutation rate, which is 3.1 into 10 to the power of 8. Means there is one mutation, 3 mutations in 10 to the power of 8 bases. Whereas you look into the mutant of DNA repair genes, you can see that the mutation rate goes up to 100, which is a difference of approximately 37-fold between wild type to the mutant. So we basically hypothesized that if you have mutations in DNA repair genes, we get higher resistance. And that seems to be the case in vitro. And none of these things are very surprising, but our hypothesis is all driven. One has to show that DNA repair genes are actually contributing factor for resistance in Mycobacterium tuberculosis. For that, in vitro, this is called in vitro when you grow things in a bacteria in a culture shaker, then it is called in vitro experiments. Then what we have to do was we did an in vivo experiment. So how do you do in vivo experiment? What we do is we take the bacteria, we culture them. After culture them, we take approximately 100 million this bacteria and we aerosolically infect guinea pigs, right? So these are done in a specialized chamber. It is done in a BSL-3. And what we do is we sacrifice or we euthanize some guinea pigs on day 1 to see if actually their lungs have taken the bacteria or not. That is called the day 1 CFUs. And after 56 days, that's almost 2 months, we look into the CFUs all over again. And we look into the survival rates of wild type and the mutant bacteria that we have generated. Right? I hope I'm clear. If I'm not clear, please stop me irrespective of where I am. So this is how a guinea pig lung looks after 56 days of infections. There will be granulomas, which may not be that apparent. For example, I can show that is a granuloma. That's a granuloma. That's a granuloma. There are granulomas. You can see here also. These are the white patches here. They are granulomas. So what we can tell you clearly is that there is a low difference between the wild type, which is this, and the double mutant. Let's not worry about the other double mutant. You can very clearly see that there is a half a log-fold difference. What is half a log-fold difference? It's approximately 5-fold difference. Means for a wild type, if I'm getting 1 colony, I'm getting 5 colonies with the mutant. That is basically what happens in case of a DNA repair mutant, which essentially tells me that my DNA repair mutant is able to survive in the mice quite effectively. So why is it surviving better? To gain insights into this, we went back to molecular biology technique. What we did is we isolated DNA from 10 different colonies, genomic DNA from 10 different colonies, and then we genome sequenced, the whole genome was sequenced for all of them. So here is what we have done. We took a guinea pig infected, we took out the lungs, from there we got the CFUs, and these CFUs are individually picked, and eventually we made a genomic DNA isolation, library preparation, and sequenced it. So essentially, bottom line, we sequenced their genomes. And what we got is, without going into the details, that here is the first one. This is the RV strain, mother strain. Mother strain, when it goes into the animal, because in the animal, we are also animals in a different kind, when a strain enters your body, it has to survive, right? To survive, it accumulates certain mutations. You can see that. The yellow is not accumulated. Some of the other colors are accumulation of the mutations. So the background is slight yellowish. That is perfectly normal. And rest of this is accumulation. So you can see that it has accumulated few mutations, but not many, not many. Now go to the extreme right, which is this one. It is a double knockout, and you can very clearly see that in case of double knockout, it has accumulated large number of mutations, and it is also reflected by its ability to survive better in guinea pigs. So what is happening is there are few mutations in the DNA repair genes that is resulting in mutations in the genome. Some of these mutations are going to be killing, are very important for the bacteria, and hence that will die. But there will be mutations which are happening in some of the other regions, and because of the selection pressure they get selected for, eventually they thrive. So essentially, having mutations in the DNA repair genes gives it a way to evolve faster. It's an evolution issue here. So it acquires mutations in multiple genes. Some of them are useful. Anything that is useful is retained. If it is not useful, they die, right? So whatever is retained over a period of time is something that is evolved compared to what original strain was. So deletions or mutations in these genes provide survival advantages. This is what we have realized after working with these DNA repair mutants. So what happens when we come, this is an incomplete sentence, mind you. What happens when wild type is competed with the mutant? See what happens in your body. Let's assume that we are infected with a tuberculosis. So now the tuberculosis went in, and let us say out of 1,000, 2 of them have gained mutations in a DNA repair gene, and that has helped them evolve antibiotic resistance. So now you have a DNA mutation, a repair mutant sitting in your thing along with 980 of the other. So now they're competing with each other for survival under that particular circumstances. Our hypothesis was the mutant, because of its ability to accumulate mutations, would outcompete the wild type and eventually become the most prominent strain in that particular niche. So to test this, what we have done is we have done an experiment in a cell line. So what you do in a cell line is we take the cell line out of mice. These are peritoneal macrophage cell lines, and we infect them either independently or together. When we infect them together, both of them are getting into the same niche. And what we do is after 36 hours, we take it out and we reinfect. We reinfect. We do this 3 times. After doing this 3 times, the strain which is thriving will outcompete the strain which is not thriving. Right? It's a very simple concept of the person who's fitter can run faster than the person who's less fitter. Right? If you're in a race. So very similar. You can see that as the time progresses, by 3rd round of infection, the mutant, or final round of infection, you can see that the mutant survives way better compared to the wild type, indicating that mutant actually outcompetes the wild type. So what does it mean? So if you by any chance, your strain, the strain in your body accumulates mutations in a DNA repair genes, when one starts treating such a TB, they have higher propensity to evolve drug resistance. So to prove this point really is true, or this is in vitro, right? What against cell lines. So to prove this point, what we have done is we have used both mice and guinea pig systems. What we did is a very simple one. We complemented it. We competed the wild type with the mutant, and we also competed in this case wild type with the mutant and wild type with the mutant, which is complemented back with those original genes. Means when you put back those genes, what happens? So to cut this story short, what I will show you is what our data is. At day one, both wild type and mutant strains behave very similarly. But as the days progress, and at around 56 days, you can very clearly see that the mutants seem to be gaining advantage even in mice infection. So this part is in the mice infection, lungs. Now we have done the same thing in guinea pig infections. You can see that by 56 days, mutant has actually gained huge advantage, this one over this one. Mutant has gained huge advantage over the wild type. However, when you put back these genes, that advantage gets nullified. So what this experiment clearly states is that mutations in DNA repair genes provided a leg up, an advantage, so that they can evolve faster. This is a way how a beijing strain may be gaining mutations very fast for any kind of drug that is given. That's our hypothesis. It's not proven. So we also had another question. If this is indeed true, if we can evolve mutations faster because of mutations in DNA repair genes, can we use this for identifying targets for the drug resistance? As a proof of concept, we used ciprofloxacin. Ciprofloxacin is a drug that acts on an enzyme called DNA gyrase, which is on this side. So DNA gyrase, as I stated earlier, is required both for DNA replication and for transcription. So if you have, so what we did is, an experiment is simple. What we took a wild type and the mutant strain and treated with ciprofloxacin, higher concentrations of ciprofloxacin. We got 100-fold higher number of colonies, or actually 400-fold higher number, 100-fold higher number of colonies in the mutant compared to the wild type. And what we did is we took 11 or 13 of these colonies and whole genome sequenced them to see where they have accumulated mutations in the double knockout, or in the DNA repair knockout. So what we got is very interesting, which is like ciprofloxacin, the target is known, and the mutations that are responsible for the clinical resistance is known. We identified those mutations very comfortably in 13 genomes that we sequenced. All 13 genomes had mutations in so-called gyrase gene, and some of the other genes were also there. It very clearly states that we can use such a strain tomorrow for an unknown antibiotic, a novel antibiotic that may be generated by any researcher. They can use our strain to identify the target for such an antibiotic. Would it matter? Yeah, because identification, quite a lot of times, how are antibiotics developed? They're developed not without actually knowing the target. They're developed where the libraries are used and things are done, right? Eventually, when they get it, for example, in case of bedaquiline, the target was not known, and they had to do in vitro evolution to find out what the target is. What we are stating is we have developed a strain which can be used to screen or find out what bedaquiline kind of drugs are targeting in the bacteria. Using this, one can do it much faster than doing the experiment with wild type. So given all these things, what we are stating eventually at the end of this, and with all the background that is given, is that if you have mutations in DNA repair genes, they evolve faster depending on the stress. If the stress is survival, they survive better. If the stress is antibiotic, they develop antibiotic resistance genes, resistant mutations. So this is what we are saying, and it is something which has recently been accepted for a publication in a place called PLOS Pathogens, a well-known journal, and that is where we kind of end the story. I have not gone very deep into the science, try to keep it superficial so that I can I mean, you guys will be with me till the end. Thank you for your attention and giving me the opportunity to talk about our work. And I'll be very happy to take any questions that you may have on any of these areas. Thank you.
Thank you so much for that wonderful talk, sir. It seemed a little difficult for me to understand a lot of, but then so many new things I learned. And then I'm trying to digest so many words, new words which were introduced. Ravi Shankar, sir, anything?
Yeah. Is there any, as we know the resistance, how it is going to develop, are there any new antibiotics are developed or are they developing regarding this one? No. Any idea?
Ravi, Ravi, that's a continuous process. There are few antibiotics that are there in stage 3. There are some in stage 2. For example, what I was talking about, bedaquiline and delamide. These are the ones which are now in market, but they were in development for quite a while, right? I mean, see, the problem is people start with libraries. Eventually, they do phase 1, phase 2, phase 3, pharmacokinetics, and all these things. It's generally a 16-year process from one end to the other. There are always a few in the pipeline. There are 2 other compounds. I don't remember their names, but they are in phase 3 of the clinical trials. If they're approved, we'll get newer drugs. But the problem is bacteria is smart. It continues to develop resistance to whatever we throw at them. And it is not a problem that is limited to tuberculosis. You are very well aware of antimicrobial resistance, which has become a major health problem at this point of time for anybody anybody who goes for any surgery, any hospital infections.
Yeah.
Like my father himself has that.
Yeah.
So it's a pretty big problem. So drug resistance is a issue. So problem is continuous development of new antibiotics. But there is one thing that I have to say, unless and until there is certain level of responsibility in taking the antibiotic course, this will continue to be a problem no matter how many antibiotics are developed. Because if doctor gives you for a 5-day, I know how many patients take from only 3 days. We know that,
Correct.
right?
Yeah, correct. Okay.
And if the 6 months, out of question, right? 3 months and you feel terrific with TB. After 3 months, they stopped it, right? These are the reasons why drug resistance comes.
So like TB, tuberculosis, any other diseases you find that it is so much complicated, so much of resistance you have been dealing with?
That's what I'm saying. Antimicrobial resistance.
Sir, Vinay, your voice is not heard.
Not.
Yeah.
Hello? Can you hear me now?
I can hear. Yeah.
So many of the other infections, hospital infections, for example, you would know that many of these infections are drug resistant to multiple antibiotics. That's because of multiple reasons, but drug resistance in bacteria per se has become the major deal. And if you really think it's not limited to India, even in places like UK, US, Europe, AMR, antimicrobial resistance, is per se considered the major problem. I mean, less about TB. I'm talking about something about completely different other bacterial infections, which have become a major issue, right? I don't remember the name of many of these organisms, but their antimicrobial resistance is a big problem across the world currently. And hospital-borne infections are the ones which are more problematic. They're already resistant and they propagate from one person to the other.
Sir, in your labs, you actually do things on this guinea pigs and rabbit is what that is what.
None of these in the lab, they are done in facilities which are for this. So I'll tell you what happens. We have something BSL-3 facility. In that, there are animal cages. So once an animal goes into that BSL-3 facility and goes into a cage, this cage is individually ventilated. Means even whatever is gone in, it's HEPA filtered, right? So once an animal is infected, we put in those cages and we do the whole experiment. And all the while, animal is in that cage in a BSL-3 facility. People enter, do the cleanup, everything, but it is completely done in a BSL-3 facility. Eventually, when the experiment is over, what we do is that we euthanize the animals, we take their organs. We do not bring anything out of that facility alive. And everything is autoclaved before they're discarded. So so that people who are outside do not get this problem. Now we work in tuberculosis. We come out and we leave it outside, and some of them has drug resistance. It can be a serious problem. Everything gets killed. Bacteria, animal, autoclaved. Everything is completely sterilized before they're discarded. Only the results come out eventually.
Okay.
That was really interesting, sir. I think we have done with the session, sir. Any?
That's all. Yeah.
Thank you so much, sir. I hope you received our token of appreciation. We would have appreciated if somebody was there to honor you there.
Thank you very much. I'm quite okay.
I know. I know his room.
Themselves.
Nobody can enter there. Okay.
We understood the situation. Thank you so much, sir. In lot of terminologies, our PGs actually have a question on recent advances and evolution of drug resistance. You explained everything very pictorially. Thank you for that lucid
It's a very new area. I'm low as in terms of basics and fundamentals, but I do know that it is still beyond a point. It becomes a little bit technical. I can't do anything about it. I have a little more technical anyway, right?
Yeah, it's okay. No problem.
I tried my best to keep it as less technical as possible.
Yes, sir. Thank you so much, sir. Thank you so much for this wonderful webinar. Thank you so much.
Thank you, Vinay. Thank you. So nice of you. Yeah. Some other time we'll have one more this one. Thank you a lot.
Yes, sir. Thank you.